This is a special episode of the Immunology podcast, Immunology 2025, Day 3. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rad. Welcome back to the Immunology podcast. We have conversations with immunologists. We're back with our third episode covering highlights from Immunology 2025 taking place in Honolulu, Hawaii. Brenda and I have had so much fun attending and getting to meet you all, but it's not over yet. If you're looking to find out what we're up to at the meeting, follow us on X at ImmunoPodcast or on Blue Sky at IMMUNOLOGY2025.com. You'll also find episodes covering the 2 previous days of the meeting at ImmunologyPodcast.com or wherever you find your podcasts. Today, we recorded a special show in front of a live audience reflecting on our insights from our past 100 episodes. The episode will air next week on May 13th. Don't miss it. We'll be discussing in a few moments our favorite sessions throughout day 3 of the meeting. But before we get to that,
working with mouse immune cells, get a free wall chart of the estimated frequencies and percentages of more than 25 immune cell types in mice. To learn more, visit stemcell.com/mouse-immune-cells.
all right, well, we had our live show, but we're not going to talk about that because it's going to be out in,
That is true, but it's great, guys. You should check it out.
You should.
It was really fun.
I will say you unleashed a dad joke pun on me. And
I did.
And my heart grew 5 sizes out of just,
I'm learning from the best.
You were inoculated.
I know.
And now your mRNA is expressing.
I'm embracing it. So yeah, but besides the live show, which talks did you attend?
I'm going to tell you about my day of mucosal immunology.
Oh, right. Everything is a mucosal immunology for you, isn't it? That's okay. That's okay.
Enterocyte is still the best immune cell.
All right, hit me. What do you want to share?
So in the morning, I looked at regulation of mucosal immunity.
Of course you did.
A couple of really cool things. So the first one was Got Milk, investigating the role of mammary IELs in promoting lactogenesis with Deepshika Ramanan from the Salk Institute. So when you're pregnant and need to grow your boobs to make milk, which is known as lactogenesis, it requires a rapid remodeling of your tissue.
Huh.
And they have found some craziness here. T cells migrate from the intestine to the mammary gland during pregnancy and early after birth, where they expand. And they have TCR, and the TCR clonotypes they find there match small intestine T cell clonotypes.
Why? Okay. All right. So is it alpha beta T cells?
Yeah. And you need microbes for it to work.
Oh my God.
So germ-free mice, this doesn't happen in.
Why?
And then they look both in mice breast milk, which I don't know how you get mouse breast milk. I have questions about the things we do for science, and human breast milk. And guess what's in breast milk?
T cells?
T cells with immune intestinal
way.
TCRs.
Really?
Uh-huh.
There's T cells in maternal milk?
Uh-huh.
Oh my God.
They come from the gut because the gut is awesome.
It makes no sense. It makes no sense, but that's okay. I guess that's how it is, but I just don't understand it. But I don't have to understand in order to be true. All right, what else? What other mind-blowing fact do you have for me?
The next one is same talk from Dayu Fang in Northwestern. Succinate drives gut inflammation by promoting FOXP3 degradation through a molecular switch. So if you add, they did a DSS model and added succinate, and that drove inflammation. There was less FOXP3 cells. They identified that it was promoting Treg IL-17 production and downregulating a couple of genes. One was DLST. If you make DLST knockout mice, it phenocopies the succinate enhancement. And then they do a bunch of stuff that they went through very quickly because people were running out of time in this session. And they basically got to the point that DLST drives a switch between succinylation and ubiquitination, because succinyl-CoA restores FOXP3 in distal DLST knockout Tregs. So succinate and succinyl-CoA are not the same, right? One comes from the other. And what they found is high succinate levels negatively correlate with FOXP3 and DLST in the blood of IBD patients. What's really happening is high succinate has Le Chatelier's principal effect and pushes things to ubiquitination and degradation. And so that then leads to the disruption of anti-inflammatory Tregs. And so high succinate pushes that, and you want more succinyl-CoA and succination, not ubiquitination. So they got that.
So of the transcription factor itself, of FOXP3 itself.
It was unclear given the speed of the talk.
Okay. Yeah, I mean, FOXP3 can be acetylated. I'm not sure. I heard it was succinylated before, but I guess why not?
Succinyl-CoA will cause the coacetyl A to move onto something. It becomes a substrate.
Okay. All right. Okay.
All right. So there were a couple other talks here. One more I wanted to talk about was the silent killer from Morgan Harwood of University of Utah, talking about how the innate immune receptor CLEC12A defends against potentially harmful microbes without causing excessive inflammation. It's a pattern recognition receptor, which is specifically a C-type lectin class, knockout subworse DSS colitis. They used germ-free mice with wild-type or knockout microbiota and found that the microbiome of the knockout transfers disease if you get rid of it, and they identified that it wasn't clearing enough F rodention, and it alters macrophage phagocytosis. So it's a important immune sensor, but it's on macrophages and driving phagocytosis, and so it doesn't cause over-inflammation when it's turboing. But if you lose it, you lose bacterial killing and sensing, and that leads to inflammation. So that was interesting. And then in the afternoon, it was the Society of Mucosal Immunology section, which had some titans speaking. And I got a list of several of them. First up was Greg Sonnenberg from the Weill Cornell Institute of Medicine, talking about innate lymphoid cell-mediated tolerance in the guts. So he's really talking about these ROR, gamma T APCs, which we've talked about on the podcast before, and a bunch of his work. So some of the papers we've talked about in the past. He was really talking about a couple things. One, they're still trying to identify all these populations. They're basically APCs that are not dendritic cells. And some are tolerant, and the more ILC3-like, the more tolerant they are. But there's some that are circulatory, inflammatory, and there's these ETAC types with these airy gene phenomenon. And they're trying to really figure out the kind of, flow cytometry taxonomy, as it were, and what their function is still. The important thing is how they respond to gut pathobionts, which is what they were talking about here, and they used Helicobacter hepaticus as a mouse model. DCs don't respond to it, but these cells do and are important for protection. And they identify C-gas and the STING pathway as being important, and that then activates CCR7-mediated migration, and that then leads to protection from the organism. They also look at an IL-10 knockout mice, which has colitis, and add this on for exacerbation of disease, and identify that while STING is good, you have more STING and less ILC3s and IBD, and they're thinking what's happening is STING overdrive leads to cell death. So usually some STING good, you're recognizing the organism, you're killing it, you're protecting the host. Too much STING, bad, right, with overdrive, and it kills the suppressive subset of these. Then you had June Round from the University of Utah talking about microbiota immune reactions that promote metabolic health. So this is a really good point that she made, which is 70% of type 1 diabetics have an HLA risk allele, but only 2 to 7% of people with that allele get type 1 diabetes. So genes are required, but environment's important. You have a loss of microbial diversity in type 1 diabetics, even early on and even with well-controlled blood sugar. And they know that disruption of the microbiome in mice, and they showed this as part of the work, early in life disrupts beta cell development. So beta cells are developed early in life, and kind of what you're baked in with beta cells when you're young is what you get for your life. And the microbiota for that matters. And they identified a bunch of bacteria in here, still exploring more, but they also identified fungi as well. And one fungi, C. this influences macrophage seeding in the beta cells, which is important for their development, and it has something to do with the cell wall. They also showed, though, that if you ablate in adult mice using, I forget the name of it, and they went really quick, but it's a canonical immune model for ablating beta cells with a chemical.
Okay.
And so it gives adult mice type 1 diabetes. But if you also throw on the C. dublinesis, you can recover those mice. They'll start popping up new beta cells, or the beta cells won't die.
Wow.
So that was really cool. And then Rodney Newberry from Washington University of St. Louis, he's the guy that discovered GAPs. So these are the goblet cell antigen pathways, which are controlled by acetylcholine. Showed that in some of the new work that if you have inhibition of the GAPs, you have loss of tolerance, which is something he'd shown previously. But he then tied it to another cell population of colonic iNKT cells. And so something they were appreciating is that these iNKT cells in the colon, which have a variety of functions, contain immature, intermediate, cycling, and mature subsets. We'd known it in mice, and he presented some data showing it existed in people. But what he really established was that the gaps are required for the antigen sensing to drive their development through CD1D expression. So it's tying in both goblet cell function to immune development. And it was pretty cool. It was a tour de force of a lot of PIs in the later afternoon for the society meeting. It was pretty fun.
Nice. Yeah, those were really cool stories. Thanks for sharing, Jason.
Will I have mucosal immunology tomorrow? We'll find out.
I mean, you should also, there's other immunology besides mucosal immunology.
I went to transplant immunology the other day.
That is true. It's just that. I mean, I'm forcing myself to not only go to T cells, although to be honest, I think.
Aren't you about to talk about T cells a bunch?
I am going to talk about T cells a bunch today, but not so much actually. All right. So my highlights of today. The first talk was the CAR T cell talk, so I'm just kind of contradicting myself. But Emma Hathaway from Vanderbilt University did a really nice study on looking at regulators of CAR T cell survival and adaptation in tumor microenvironments. That's the title of her talk. And basically, she compared, we know that CAR T cells are, not really up to the challenges for solid tumors, and we're still trying to understand the reasons why. And so she basically, I think that her experience were pretty nice in the sense that she kind of went back to the basics. She compared models in which she had both CAR T cells and TCR transgenic T cells targeting antigens in the same tumor. So basically, the experiment, she had like a tumor that had was a mix of two different clones. One had a CAR T cell target, one had a TCR target. And then basically she inoculated mice. Inoculated mice with these tumors, and then she transferred T cells. Half of them were CAR T cells, and the other half were OT1 cells. So the TCR antigen was OVA antigen. And basically, she just like what you see is that the CAR T cell, the OT1 part is much more efficient than the CAR T cell part, really suggesting that although at the beginning the TCR transgenic T cells are a bit slower to start, in the end they are the ones that are left standing, and you basically don't see any CAR T cells anymore in that tumor. And so trying to understand what are the genes that are to some extent mediating the cell death that you see in the CAR T cells. She did a in vivo CRISPR screen, and surprisingly, one of the hits she saw was the IL-2 receptor alpha gene, better known as CD25, which is the subunit of the IL-2 receptor that has the kind of the highest affinity and gives the whole complex a high affinity for IL-2. And so what she sees basically that if she knocks out IL-2 receptor alpha, the knockout cells accumulate in the tumor much more efficiently than cells that are expressing IL-2 receptor alpha. And so basically, there's a disadvantage when these cells are growing in vitro. They have a disadvantage because they are growing the IL-2. But they have a growth advantage once they are transferred to the mice adaptively. So she like this kind of the observation that they see. It was a short talk, so she didn't go much further than that. But I thought it was kind of cool because a bit unexpected. But when there were some comments made that, well, it is well known in the adoptive cell therapy field that growing T cells in high IL-2, which is how most of the, for example, the tumor infiltrating lymphocyte protocols are. The TIL production depends on really high amount of IL-2. And then when you transfer those cells to patients, you actually have to also administer a fairly high amount of IL-2 to the patients in order to keep those T cells alive. And that's a problem because that brings some toxicity. So like most of the toxicity from TIL therapy comes from the IL-2. But so I guess that this makes sense that there's this idea of like you've made your T cells addicted to IL-2. So if you can prevent that, that might help in the long term. Maybe not in vitro expansion, but maybe in the long term for persistence. in a situation you might not have IL-2 around. So after we had a talk by Jose Conejo Garcia from Duke University, which on a kind of a side note, it was a bit sad because it's actually two postdocs from his lab were supposed to attend and give the talks. Unfortunately, given the current immigration policies, these are people on visas, and they did kind of did not feel comfortable flying at this day in this time. So he took over one of the talks. But yeah, that kind of sad byproduct of our current situation. But I like this because this was about gamma delta T cells and the potential of using gamma delta T cells for cell therapy. And basically CAR T gamma delta cells. I thought that was pretty cool. So he talked a bit about the differences between gamma delta T cells and alpha beta T cells, and they're quite different cells. Primarily, I think when it comes to gamma delta T cells, there's a limited combination of gamma and delta chains that combine. So it's not like alpha beta T cell, which you have this massive diversity, but gamma delta cells are a bit more concise. And but we don't exactly we know some of the targets of gamma delta TCRs, but not all of them. And so he really kind of wanted to make this point that there's still a lot we don't know about what gamma deltas can recognize. So he looked at gamma deltas in high grade serous ovarian carcinoma. And basically, what he saw when he characterized this subset is that there is a dominance of gamma delta T cells that have a V is a delta chain that's this V delta 1 and gamma 9. So this combination is very prominent. So brings the question, what is it about this receptor, and what is the role of this receptor in gamma delta T cells? They also see other chains, but basically he characterized the type of gamma delta populations that you see. And they're different populations can be identified with different kind of trajectories of differentiation when he looked at the overall phenotype of these cells. But in general, what is interesting is that they seem to be more resilient, more resistant to exhaustion than alpha beta T cells in the same tumor. When you're looking at kind of classical exhaustion markers. And the point, so I think more translational part of the end of his talk is using gamma delta T cells for targeting, for therapy, and particularly making CAR gamma delta T cells from umbilical cord. So gamma delta T cells isolated from umbilical cord, which are fairly abundant compared to more adult tissues, and that you can actually, get these cells, transduce them with a CAR, expand them. The protocol is a bit, they needed to optimize the protocol. Particularly IL-7 requirement is very important for gamma delta T cells. Basically you can have these off-the-shelf products that is gamma delta T cells from just umbilical cord, and they seem very promising as a adoptive therapy for cancer because of these different performance in tumors. And another interesting kind of new approach for cell therapy was from Megan Pearl from UC Davis. And basically she looked into placenta derived NK cells. And getting NK cells is not always easy. They're not that abundant in blood, but actually you can get a very nice population with very good characteristics from placenta, which is basically an organ that is discarded after birth. And this is also another idea of allogeneic cell therapy style. And she showed that these cells interestingly have a higher expression of CD16 than NK cells derived from PBMCs. And if you combine this with what they show is a tri-specific killer engagers that are recognizing B7H3 on tumors, and they have this anti-CD16 that binds to the CD16 on NK cells, recombinant human IL-15 module that is for stimulating the NK cells, and then the short chain fragment from the anti B7H3, and that this combination actually works really well for targeting tumors. They were looking into leiomyosarcoma, but I guess they also would apply for other tumors. So this combination of placental and NK cells with this trispecific engager was really a really winning combination. So a little bit, of unorthodox approaches. And at the afternoon, I gave myself a treat and I attended some of the talk from the veterinary and comparative immunology. And you know what was the first talk I want to highlight? What was it about?
Was it sharks?
Well, it was sharks. It was sharks. Ah, it was great. So basically, this is a talk from Thomas Hill at the University of Maryland, and um, yeah, sharks have a surprisingly complex adaptive immune system. Is the oldest organism that has an IgG, an immunoglobulin based adaptive immune system. So they have B cells that make antibodies, and they, the antibodies that are maturing and producing IgG. And so always the question is, where are these antibodies made? And so basically, the talk is called The Unexpected Role of the Nurse Shark Pancreas as a Secondary Immune Organ. So that kind of gives away what I'm going to talk about. So I think that the kind of the idea started when you see, stains of the shark pancreas. You see the structures that look suspiciously like B cell follicles that you see already in the spleen. That has been shown that they have it in the spleen of the sharks. And so he's like, well, they look a lot like B cell follicles. Is a community. So let's just do some staining, some analysis. Turns out there are indeed B cells. They use IgM. They can find these follicles, this clustering of IgM indicating these follicles. Comparative that can be compared to those ones in spleen. They're like nested within the pancreas, physically separated, like clustered away from those cells that actually make classical pancreatic proteins like amylase or insulin, but they're still there, right? They just find these little hot spots of B cells, and they show that indeed those spots, they take up antigen. They are B cells are proliferating, are mutating, are expressing Ki-67, 8, and their T cells are associated with these follicles, and there you can actually find mature B cells that are producing IgGs from the pancreas. So there's like a backup spleen in the pancreas of these nurse sharks. I thought it was pretty cool. And then also in the same session, studying the immune compartment of elephants. Elephants are really cool. These huge beasts. They live very long. They don't get cancer, and so they were looking into the PBMCs of elephants. And one thing that kind of pathologists that look animal pathologists that look at like slides from blood from elephants, they notice very quickly that they have a lot of monocytes or things that look like monocytes, and that has been kind of known at that kind of very superficial level. So they actually did some sequencing of cells, and they show that indeed they have like 50% of the PBMCs are monocytes compared to humans. There's like about 5%, so they have 10 times more monocytes than humans do. And it's not clear why they're there, but that's how they are. They have this huge population of monocytes.
Do they eat the cancer?
I don't know. They didn't look into that, but maybe.
Well, you did pretty good. It was only 2 out of like 5 or 6 that were T cells today.
Yeah, that's true. See, I have elephant monocytes. What more?
Well, I didn't expect you to be in the vets in the afternoon.
Doesn't get more exotic than that. And I just want to say, then I'm gonna switch. For the last talk, I went to a different talk. So I just want to highlight, this is from our friend of the podcast, Leo Ferreira, and his student, Russell Wayne, and Cochrane, Russell Wayne Cochrane, I guess that's his last name. And basically looking into T-Rex, and what is observed that expression of CARs on T-Rex sometimes can lead to kind of differentiation of T-Rex phenotype. So they showed that, and at least for the CARs that they tested, that the T-Rex could end up producing pro-inflammatory cytokines, particularly interferon gamma, not IL-2, which is good because that would really mess up a T-Rex phenotype, but interferon gamma, and CD40L, which is mostly reserved for effector T cells, so really a move away from Treg phenotype, and that this, to some extent, was due to the high affinity of like high affinity CARs did that more than if you have a low affinity CAR, you can kind of prevent that. And they show some mechanism around behind that. They show that there's this protein called IER3, one of the top genes correlated with this pro-inflammatory Treg phenotype caused by the CARs, that is downstream of NF-kappa B, and that's a negative feedback on NF-kappa B. So they show the overexpression of this IER3 actually leads to lower expansion of Tregs in vitro, but improved CAR Treg suppression capacity and prevents this acquisition of interferon gamma production and this kind of the loss of Treg function identity, so to say.
So maybe quality over quantity.
I think so. Yeah, I think that's a kind of a theme that I see more and more in the T cell field. The Goldilocks zone. It's really important. It cannot be too much or too little.
More is not better, as we know from autoimmunity.
Indeed. So that was fun.
All right. Well, that brings us to the end of our latest Immunology 2025 episode. Don't forget to follow us on X at ImmunoPodcast or Blue Sky at ImmunologyPodcast.com to find out what we're up to at the meeting, the ridiculous shirts that I may be wearing, and check back here tomorrow for another episode recapping day 4 of the meeting. See you then.